The idea of terminating a fetus which is recognisably human upsets some people. But genetic medicine has found a way to test for the genes that carry disease much earlier in the development of a human life. In fact, so early that the fertilised egg has not even implanted in the womb. In vitro fertilisation, or IVF, is a tried and tested fertility treatment where the woman's egg and the man's sperm come together in the laboratory rather than in the woman. In normal IVF, doctors grow the fertilised eggs for 4 or 5 days, then implant them into the womb. But with PGD, or pre-implantation genetic diagnosis, they test them first. The embryos grow in the laboratory for 3 days, until they're 8 to 16 cells in size. Then, 1 cell from each embryo is carefully removed.
And then the process is very carefully to make a hole in the shell of the egg, grasp one of those cells, take it out, and use that for testing, and the rest of the embryo is put back in the incubator. So very, very gently coax it out. And this is quite delicate because obviously you can break the cell very easily.
Amazingly, this procedure does not kill the embryo.
So it gently brings it to where the hole is. There you are, gently brings it against the cell. Now she'll put a tiny bit of vacuum on that cell, and there it comes out through the shell. And that will now be spat out of the pipette and passed over for collection for analysis.
This tiny cell is then taken to the lab, where scientists check the gene sequence for signs of SMA. The computer reads and translates the DNA sequence of the gene into a graph. Just one mistake in the line can be enough to indicate the disease.
And then we have an answer the next day and have to make a decision about putting back 1 or 2 embryos that were unaffected by the disorder.
The advantage of that is that the couple, right from the beginning of the pregnancy, if the fertilised egg does establish itself, will know that that pregnancy is at very low risk of being affected.
Which we didn't think for a couple of weeks was right for us, but after we'd sat down and talked about it and decided that it would be something that we'd like to go down really, to see what, what could be done for us.
And so it was that 3 years ago, Tina decided to try pre-implantation genetic diagnosis to conceive a child without SMA.
Here it comes.
While she was not prepared to terminate a natural pregnancy, she was prepared to choose which balls of cells in the laboratory should have a chance of life and which should not.
Well, for me personally, I think once the eggs have been, not so much that they've been fertilised in the test tube when we had the PGD, I don't feel that it would have mattered as much then if they'd not survived or whatever. But once it had been implanted, I feel that was a point where that's where life begins, as far as I'm concerned personally. I feel that I would have then become had a bond with that child, or children as in our case. That's at that point that I would have felt it was a life.
PGD treatment usually has a 1 in 5 success rate. So it was a particularly happy day for the Snapes when their twins, Esther and Isabel, were born.
I was always desperate to have some sisters or brothers. At first I wanted a sister, but I got 2 instead of 1.